Good morning, class. I'd like to start our lecture on hemostasis. Don't confuse hemostasis with homeostasis. Of course, homeostasis refers to a stable internal balance. Hemostasis refers to the formation of a clot so that you stop the flow of blood. And also, hemostasis should not be confused with the with bleeding time. Clotting times and bleeding times are totally different things. And I will talk about that in class. So this is from chapter 36 of Guyton and Hall, and we have a lot to talk about. The last part of our lecture will deal with manipulating hemostasis. That's your clinical application. I'll talk about heparin drips and Coumadin and what happens to clotting times. Do they increase or decrease? Aspirin, it's an antiplatelet. So I will talk about all those things, and then we will also talk about, towards the end, problems with hemostasis. Patients who have a tendency to clot or patients who have a difficult time forming a clot in the first place. Speaking of which, our hemostasis balance must also have homeostasis. It must have a balance. Now, we need a balance between factors that promote coagulation. These are called procoagulants. And we need a balance with the anticoagulants, naturally endogenous substances that promote good vascular flow. Now, we normally associate our blood as being a liquid. So obviously the anticoagulants are, for the majority of our lifespan, are the dominant factors, right? We want our blood to be in this liquid phase and flowing through our blood vessels. However, when those blood vessels are torn or compromised in any way, we would like our clotting factors and our procoagulants, if you will, to now be dominant and form the clot. As I alluded to in the slide previous, I said that I would talk about people who have inappropriate clotting and people who fail to clot. So I will talk about people with thrombocytopenia, I'll talk about hemophilia, and I'll talk about people who have a tendency to form clots too frequently. Now, the tendency to form clots, a clot is a thrombus, and when they are mobile, they are an embolus. And I will tell you in the later segment how to remember the difference between those. So that would be associated with inappropriate clotting. And then if we tip the scale the other way and we can't form a clot, obviously we're going to have a problem with bleeding out into our tissues. So I'll talk about the different types of hemophilia. Starting right now, you should start making a list of all of the endogenous anticoagulants and procoagulants. And by endogenous, I mean coming from within the body, of course. So just start making a list of what you hear me talk about that would either favor blood clot formation or would inhibit it. Let's start with platelets. Platelets are part of the clot formation, so they would be the first one on your list for procoagulants. Now, platelets are not true cells. They are membranous sacs of clotting factors. They come from a larger cell called a megakaryocyte, a very large cell. Megakaryocytes, if you listen to an earlier lecture, come from a mother progenitor stem cell, the hemocytoblast. And you might remember me saying that when it replicates and divides, the daughter cells can choose the pathway or are induced into a pathway to differentiate, completely change their form into mature cells that are found in the blood, either red blood cells or all the different types of white blood cells. Well, one of the pathways that the daughter cells can go into is for thrombocyte formation. And the megakaryocyte is the cell that will bleb off parts of its cell membrane within it parts of its cytoplasmic contents. So these platelets, you have about 150 to 200,000 of them per microliter of whole blood, and these platelets have a lifespan of about 8 to 12 days before they are removed from circulation by the liver or spleen. Now let's talk about what they have inside them. Guyton and Hall talks about the different types of granules, and I don't want you to worry about that. I just want you to know what we find in a platelet. We do find organelles. They do have mitochondria. The mitochondria are functional, so they are still making ATP, right, the cellular form of energy. And with hydrolysis of a phosphate, then we would have ADP, so ATP adenosine triphosphate. If we break off one of the phosphates, we get ADP, adenosine diphosphate. And now I want you to put a star by ADP because that's going to promote clot formation. There are also other organelles like endoplasmic reticulum and the Golgi apparatus. And these are going to help make some of the clotting enzymes and also provide a way to store calcium. Calcium is a cofactor that is required for clot formation. Without it, clotting times increase, meaning it takes longer to clot. There are also prostaglandins, and I want to remind you from our thermoregulation lecture that there are many different types of prostaglandins found synthesized throughout our body. They are all synthesized from a cell membrane phospholipid, and the fatty acid tail that is cut off is arachidonic acid. In this case, prostaglandins, the type that are found in platelets, is thromboxane A2, which is abbreviated as TXA2. We also find contractile proteins like actin and myosin, and this is going to help in clot retraction. You know, when the clot is actually formed and the two ends of the severed blood vessel need to be brought closer in proximity so that the vessel wall can be repaired, it's actin and myosin that are doing the contraction. So you don't find those things just in your skeletal muscle or cardiac muscle or smooth muscle. We also find some clotting factors. Not all of them are found in the platelets, but factor 13, fibrin stabilizing factor, is found within the platelets. And also there are different types of cytokines, chemical signals that will target other cells, and the type of cytokine is that is released by the platelets is growth factor that will help the endothelial lining to be repaired and the vessel wall integrity restored. Just another quick thing before I move on. The hormone thrombopoietin, very similar to erythropoietin that I told you about. Thrombopoietin is made by the liver and kidney and stimulates the bone marrow to have the hemocytoblast increase in its rate of mitotic division, and also promotes more of the daughter cells to go into the megakaryocyte line of differentiation to promote more platelet synthesis. Now, just a quick picture. This is the lineage of all of your blood cells. On the left here in gray, you are seeing the mama progenitor stem cell, the hemocytoblast, and it replicates and it divides. Now, the initial daughter cells will choose a general pathway. I say they choose, but it's really chemical signals that will induce them into different pathways. Now, the two broad categories are either the lymphoid stem cell colony, or CFUL, that stands for Colony Formation Unit Lymphoid, or the myeloid colony. And if you look at the lymphoid lineage, you'll see that the lymphoid stem cell can differentiate into either your T cells or your B cells, known as your lymphocytes. The myeloid stem cell has many different lineages, and daughter cells that are provoked into those lineages will eventually differentiate into all of your other white blood cells and your red blood cell and platelets. So we see eosinophils, basophils, the monocytes, of course, that turn into macrophages when they are fully mature. And down towards the bottom, on top of the red blood cell, you see the lineage for the megakaryocyte, and I'm showing you the little tiny platelets that bleb off of them. So the megakaryocyte is the differentiated cell that generates platelets, but the megakaryocyte comes from ultimately the mama stem cell known as the hemocytoblast. Now, the act of hemostasis involves 5 main phases. The act of hemostasis is going to prevent blood loss through damaged blood vessels. It requires the formation of a clot, and it will establish a framework for the blood vessel to repair that tissue. The 5 main phases are, first, a vascular phase. This is where the blood vessel vasoconstricts and will decrease the amount of blood that is leaking through that torn or damaged vessel. Number 2, platelet plug formation. Students have the misconception that this is the actual clot. It is not. A platelet plug is not a clot. It is just a loose assembly of platelets. Phase 3 is the actual clot formation phase, the coagulation phase. Phase 4, clot retraction, where the torn ends of the vessel are pulled together through actin and myosin interaction to allow for proper vessel repair. And then the last phase is the actual repair of the vessel. And then once the vessel is repaired, of course, we don't need that clot anymore. And so the clot should go through dissolution, basically removal of the clot. Let's go through each of the 5 phases step by step. First, the vascular phase. Now, when you cut through a blood vessel, maybe you've cut yourself with a knife on accident, the blood vessel has a natural endogenous response, a local response. And this is a myogenic response. Myo means muscle. So the smooth muscle that's found in the tunica media naturally has a spasm. The actin and myosin interact and we get the vasoconstriction. We associate this with a smaller diameter, as shown here in the bottom right picture. And this, of course, is going to reduce the amount of blood loss through that vessel. So the natural smooth muscle contraction leads to a decrease in blood vessel diameter, and this leads to less blood flow out of that damaged vessel. Now, this process is enhanced by a prostaglandin, thromboxane A2. Thromboxane A2 enhances the vasoconstriction of these blood vessels. There are other local factors, cytokines, that are acting as autocoids. That just means the cell that releases them also responds to it. Don't fret over that term. And there are also nerves that can augment this pathway. But the take-home message is that the blood vessel diameter decreases in size. Let's move on to the platelet phase. Now, the platelet plug formation, like I said before, is not a clot. What we have are platelets that come into contact with the collagen threads. If a blood vessel is torn, the endothelial lining is compromised, and the platelets come into contact with the collagen threads, and they are going to swell, they are going to change their shape, and they will release their granular contents. So the platelets are normally repelled away from the endothelial lining of a blood vessel because of a natural anticoagulant, a different type of prostaglandin that is made by the endothelial lining. This is called prostacyclin. But when the endothelial lining is compromised and the platelets come into contact with the collagen threads in the tunica externa or adventitia, then these platelets will swell in size. They get very irregularly shaped. They look like they have little feet. These are called pseudopods. and the platelets start to release their granular contents. They start releasing ADP and thromboxane A2. And to remind you, thromboxane A2 is going to help augment the vascular phase that I just told you about. All of this happens very, very quickly, within 15 seconds of injury. Don't memorize the time factor for any of these steps that I'm going to tell you about. I just tell you about the time because I want you to know that this is a very fast process. Now, again, the platelets, they come into contact with the compromised endothelial wall, the basement membrane is exposed, the collagen fibers stick out, and the platelets start to insert little receptors in their cell membrane. That's why they look like they have feet. It's really receptors. And these receptors are going to interact with von Willebrand factor. Von Willebrand factor is a protein, and it's responsible to shuttle around factor VIII. So it's found in the blood. It's a plasma protein, and its job is not only to allow platelets to start grouping together, but it also is responsible for bringing factor VIII. And people with a deficiency in von Willebrand factor have a unique presentation, they are very similar to a hemophiliac, but it's not because they lack a clotting factor, it's because they lack the carrier protein for the clotting factor. And I'll talk more about that later. So these platelets swell, they insert their receptors. It makes them look like they have little feet, and I'm going to show you a picture of this. And when they insert these receptors, they become sticky. It's not that they're literally sticky, it's now they have their receptors and they start grouping together. And again, I'll show you a picture of this. And it turns out that when a platelet is activated, so when platelets get activated and they insert their receptors, like I was saying, it causes them to start clumping together. This is still not a clot, and they are basically forming a mound of platelets. So this picture of the pills down here at the bottom right, or the beans I should say, let that represent the platelet plug, a loose association of platelets, and they are dumping out their granular contents. And part of the things that they are dumping out and releasing consists of ADP, and ADP is going to stimulate other platelets arriving at the scene to start to become activated, to change their size and shape and insert their receptors for the von Willebrand factor. So this is really a positive feedback step where activated platelets lead to more activated platelets. It is an amplification effect. So the release of ADP causes other platelets to become activated, they insert their receptors, and the mound of platelets grows. Thromboxane A2 is also found in the granular contents, like I said, and causes more platelet aggregation, but also influences the local vascular response, leads to vasoconstriction, as I mentioned in step 1. The platelets also release calcium, and calcium is a cofactor for the clotting process to occur. What is the difference between a cofactor and a coenzyme? Both help enzymes do their job, make them more efficient, but a cofactor is an element. We can look to the periodic table of the elements in our classroom, and I can point out many cofactors. Calcium's a big one that's used in the body, but it's not the only. Magnesium is another example. A coenzyme is an organic molecule. It's carbon-based. So a coenzyme is a larger complex. A cofactor is an actual atom or a molecule of atoms like calcium, magnesium, etc. Mounds of platelets are going to start, or this mound of platelets will grow. It is the platelet plug. It is not firmly held in place, it is a loose association, and this platelet plug can dislodge, and the blood can then leak out of the vessel again. The clot is actually the fibrin threads that form and weave through each other that forms the plug, or sorry, the clot, I should say, and will trap the platelets in this plug at the site then. But until that fibrin meshwork is in place, the platelet plug can dislodge. So here is a picture of a blood vessel that has been severed, and you could see here in step 3, look up there where it says number 3, you can see the disc-like shape of platelets when they're not activated. When they come into contact with exposed collagen fibers in the basement membrane or in the tunica externa even, you see the platelets change their size and shape. So they swell and they become, they look like little stars in this picture. Those little star-like pointed areas are called pseudopods, and they look like this because they're actually inserting receptors. The receptors are going to allow them to interact with that plasma protein, the von Willebrand factor, and they're going to start stacking together and form a platelet plug. Now if you look over towards the left, you'll see an intact blood vessel, healthy endothelial cells. They are releasing a prostaglandin called prostacyclin. And this is the typical. This should repel platelets so that they don't come into contact and become activated. So that prostacyclin is a very important endogenous anticoagulant for you. It's basically inhibiting some of the earlier steps of hemostasis. Don't get those platelets activated aberrantly. You only want them activated if there is a breach in the blood vessel wall. So this picture here is another way of showing you how the platelets can start aggregating together to form the platelet plug. And what you're seeing in the top right picture is a blood vessel, shown here in red. It's torn. You see collagen fibers that are now exposed, and the platelets now are going to come into contact with the collagen fiber, and they are going to insert receptors, shown here as little black boxes. And you see the little green star, and that's the von Willebrand factor. And you could see how von Willebrand factor is basically allowing those receptors to interact with it, and then it can start grouping with many different platelets to help form the platelet plug. And in the bottom right picture, you're seeing another representation of a torn blood vessel. The little white square boxes represent endothelial cells. You'll see in the middle of the picture that they are not there. Collagen fibers are now exposed, and you see the disc-like platelets that are not activated coming into contact, and then all of a sudden they get their little star kind of size and shape with their little pointy ends. That represents the receptors being inserted, and now they start linking together and form the platelet plug. So again, this is a positive feedback mechanism where activated platelets lead to release of chemical signals to lead to more platelets being activated. So what can activate a platelet to swell and insert its receptors? Obviously the collagen threads, or fibers I should say, in the basement membrane or tunica externa. Activated platelets can lead to activation of platelets. The release of ADP from the activated platelets, and ADP can come from other sources as well, say damaged red blood cells. Thromboxane A2 helps in the activation of platelets. But there can be other activators of platelets. For example, epinephrine. It used to be that heart attacks were considered to be a man's disease, quote unquote, and the men back in the day were the ones that would go to work and have the high stress jobs, and the constant release of epinephrine from dealing with that stressing job. and then that would promote their platelets to become activated and lead to ultimately aberrant clot formation, and they would, have the heart attack at their job. But that's no longer the case. We certainly have women now in the workforce. We have women that are doing high stress jobs. So anyway, excessive epinephrine can also lead to platelet activation. Thrombin is another factor that can lead to the activation of platelets. Thrombin is going to be part of the clotting phase. So we're going to see factors in the later stages of hemostasis that can basically come back to the earlier phases of hemostasis and act and promote more synthesis. So it's positive feedback galore throughout this entire process. So ultimately, at some point in this lecture, you should be getting very frightened because all you're seeing is positive feedback, and you heard me say the first day that all positive feedback mechanisms need to be ultimately stopped by a negative feedback. So you should be getting nervous and thinking to yourself, when are we going to get the negative feedback to stop this? Because I don't want all of my blood to turn into this gel-like state. Well, I will get there. So in our next segment, I will start with the third step of hemostasis, the coagulation phase.